Thank you very much for the lovely introduction, and thank you very much for the opportunity to speak to you today about some of our results. This was a project that was set up with a collaborative project that was set up with the UAE Ministry of Health as a pilot study to do, among a number of things, whole genome sequencing. I just want to give you a little bit of background, and I won't delay on this because you've given me a lovely introduction. I'm from Ireland, where we get virtually no sunshine whatsoever, and you know, just to kind of cover everything, I've been working in an Irish hospital for many, many years, and in 2016 I moved to Dubai, and you might ask, why did I move to Dubai? Well, the reason is quite simple. I came on holidays, and one of my colleagues, my ex-colleague, said, can you come and have a little look at this lab? It's really, really nice. It's really fabulous. And I was blown away. I can honestly say the facility was amazing. The structure, the design, the layout, how everything was handled throughout the laboratory was really phenomenal. The technology that they had was the most advanced technology. In Ireland, we would have been scrounging for funding to be able to get some of the facilities that they had there. The quality and the standards within the laboratory were really high. You know, the vast majority of their assays are accredited, ISO accredited, and the range of services and what you can do within the laboratory was huge. So the capacity of what we would have been able to do was great. And the staff are highly qualified. So, you know, 40% of the staff have PhDs or much, much higher qualifications. So when I was asked, you know, would you like to come and develop our NGS Based Service? Absolutely, yes. I was very, very interested. So what have we done over the last 2 years since I moved here? We've set up a range of services from genome, exome sequencing, also a range of clinical targeted panels. There's over 26 different targeted panels, and these are fully customizable. So, you know, clinicians can ask us which of the, you know, roughly around 5,000 genes, they can cherry pick which genes that they're interested in. We also have cancer panels along with microbial analysis. Microbial analysis is a metagenomic sequencing using either 16S or shotgun. So we have a huge array of stuff that we can do. On top of all of this is a comprehensive service. So it's not just the wet lab that we can do. We have 2 bioinformaticians in-house, and they can do all of the bioinformatics, and we have an interpretation and prioritization and a reporting structure in place. You know, clearly I'm not happy to put my name on a report without extensive validation in place, and the vast majority of what we spent doing over the last 2 years has been validation. And the validation that we've done has been according to the next stock guidelines, which were established by the CDC and in affiliation with CLIA, the accreditation body in the States. And it was along kind of a 3-tiered system that we use. So the next stock guidelines they cover, you know, everything from sort of the clinical, the whole clinical practice right through to the pipelines and looking at those. So the approach that we took was a 3-tiered approach. We looked at the wet lab. We did an awful lot of validation. I'm really sorry. Do you mind if I can see? Thanks. With accuracy, repeatability, reproducibility, sensitivity, and comparison to standards. We also did extensive validation of our pipelines. These are in-house pipelines, and we used in silico data. So you know exactly what this data is, and you know what you're supposed to be getting out of it. And then we validated the whole process, in particular using samples from the Genome in a Bottle Consortium. These are very, very well standardized samples with the NIST in the States. So our system was in place, and it was on this basis that the Ministry of Health contacted us in order to do this pilot study. And they were interested in a range of our different services, from genome sequencing, exome sequencing was stuff that we've done later, and also our clinical targeted panels looking for diabetes, and also the comprehensive cancer panel for inherited germline disorders within a very strict turnaround time. We managed to get all of this work done within 5 weeks. We gave full bioinformatics analysis and a report. Clearly, in the interests of time, all I'm only going to be able to talk to you about today is our genome analysis. So what was done? Basically, the idea of the project was a proof of principle in order to see, can we actually perform whole genome sequencing here in the UAE? to establish what are the variants prevalent within the local population, and also what are the comparative genomics, so what's unique to within this population, the Arabic population. There was 3 samples analysed for whole genome analysis. There was 3 separate individuals that consented to the whole genome sequencing, shallow sequencing. And the actual work involved in this, the library preparation, is very basic. In actual fact, it was the TrueSeq DNA PCR-free. It just chops everything up, adds adapters, normalizes, and sequences using the NextSeq 500. The bioinformatics, as I've said, has all been done in-house with our 2 bioinformaticians, and the variants were called using the Platypus tool, and we eventually evaluated the phenotype using Clinvar. What did we find? They are moving. So basically, we had roughly around 650 million reads that we observed, and out of those we had around 5 million reads, which were 5 million variants from that. The vast majority, 99% of our variants were single nucleotide variants, with 0.05% being indel variants. What was happening between the 3 different patients? So when we looked at them, in actual fact, you could find that there was 177,000 variants that were common amongst the 3 individuals. And what does this mean? So 9.9% of our variants were present in all 3 patients, or all 3 individuals, and about 35% of them were present in at least 2. How does this compare to what's happening with other public databases? So we looked at what we called de novo variants, so they were not present in any of these other databases, and we found that there was over 300,000 variants, so which was 5.7% of our total variants observed, that we called de novo. And again, how does this compare amongst the 3 individuals? We actually found that 6,000 of the variants were de novo on public databases, which were found in all 3 variants. So this is really important. They've previously been unreported. They're common to all 3 separate individuals, and yet they're variants that very much indicate that they might be specific to the local population. Where were they localized? So as you can imagine, 97% of them were localized in intergenic, intronic, and non-coding RNA regions, with 0.3% being located within the exon. And this is clearly not that unusual because, as we heard about earlier, 1% of the whole genome is actually the exome. But it's a really, really important little 1%. This is where the vast majority of germline disease-causing mutations are located. When we compared the results to Clinvar in order to determine the phenotypic evaluation, you can see that Clinvar has roughly around 151,000 clinically relevant phenotypic variants. And when we looked at it, the vast majority of our variants are variants of unknown significance. And only just over 3,000 of our variants were on Clinvar itself. So this really highlights the need within the Arabic population to be able to collect both genotypic and phenotypic information in order to try and drive, as there was discussions yesterday, make all of these variants of unknown significance into, can we get a diagnosis in a way that we can call them benign and we can move forward in order to help to provide a better answer for clinicians and their patients in further assessments. Of interest, 2 of the patients had a variant within COL4A4 gene, and this is a variant associated with Alport syndrome. This was originally reported as benign on Clinvar, but in actual fact, it has been upgraded by the Saudi King Faisal Specialist Hospital and Research Group to be, in 2015, to be likely pathogenic. And this means that this variant, as was discussed very well yesterday, this variant may quite likely have a deleterious effect which is specific to the Arabic population and needs to be analysed. So it was really hard to draw a conclusion from 3 genomes, I'm going to be honest. But overall, it was a really successful collaborative project, the pilot project between the Ministry of Health and the MBG Lab. We successfully managed to use NGS technology in order to be able to do whole genome sequencing and establish a local database of variants. The variants we definitely found variants that were specific to the local population. There's definitely a need in order to collect both genotypic and phenotypic information within the Arabic population. And the overall aim of this project is to lay the foundation for personalized medicine in the UAE. And the ultimate aim is to lay the foundation to predict disease susceptibility for clinicians and to guide therapeutic intervention. So what are the future expectations for the Middle East in general? And you know, I listened to a lot of the talks yesterday, and I can honestly say we had some lovely talks. Professor Al-Kharaya gave a lovely talk about how we can use autozygosity not only to help other databases to improve what they have, but also to be able to identify what's happening within our own population here in the Middle East. We had a lovely talk from Emma Babel about Genomics England, and you know, I had a really good chat with Emma afterwards, and I can honestly tell you, apart from the wealth of information that they're getting out of this 100,000 genomes, the end result of this project is that they have ended up with a standardized genomic testing across the UK. And it doesn't matter where you are within the NHS, you're going to have the service irrespective of whether you're beside the specialist centre or you're not. And this infrastructure will be in place for years to come as a result of this project. I was really saddened yesterday. I talked to a number of people and they basically said, we're trying to establish our own NGS service. And that's fabulous. I think it's really, really great. There's a lot of money being spent on it. Let's collaborate. Let's make it. Let's spend the money together. Let's make it into a service that's actually going to go forward and develop an infrastructure within the Middle East that will help clinicians and ultimately help your patients. I also talked to a lot of people and they all said to me, we send about 1,000 samples a year abroad. This is not benefiting you. This is not benefiting the Middle East. This is not getting you an infrastructure in place which you can develop from years to come and get you something that will help clinicians and ultimately help your patients. So I personally feel CAGS is very, very beautifully positioned to be able to amalgamate all of this information together in a database. The GME Variome is the start of all of this. I strongly, strongly feel we should definitely collaborate. Let's try and make this into something that can work in the future. And no matter where you are, there's politics. Let's overcome it. Let's try and get something that's going to work well together. I'd like to give a special acknowledgement, I suppose, to the project coordinators in the Ministry of Health, Dr. Kaltoum Mohammed, Dr. Noor Majid Al-Mahiri, and Dr. Mohammed Novid. And also, you know, clearly this is not my work. This is an entire team. Everybody in the MBG Laboratory was very, very instrumental in making this work. And I suppose a special thank you to Dr. Kamal Kanazari, because I wouldn't be here without him. I wouldn't be enjoying this sunshine. I'd still be in Ireland in that horrible wet weather. So overall, a very, very special thank you to everybody in the team as well. Thank you very much. Any questions?